Hello students, welcome to this amazing lesson in which now before moving ahead to the actual process of cloning and how it works, let's have a look at what are the features which are required by the cloning vector to perform the function efficiently. So let's have a look at features required by the vector to perform cloning. Features which are required to be a good cloning vector. Cloning vector. Correct? Basically, when we talk about the bacterial vectors, yes, those vectors which will be inserting into bacterium and then getting a product, we have three major requirements. First requirement is the presence of ori region. Yes, ori stands for origin region. So, the vector which you are supposedly inserting in the bacterial cell, that should have a good ori region. Yes, the site from where replication will take place. So, the complete plasmid or the complete vector will have a kind of sequence from where the DNA replication process machinery will understand that now I have to start the replication here. So, that becomes my first requirement. Second requirement is selectable marker. Yes. Selectable marker. What could be this now? Basically, if I insert the desired DNA in the cloning vector, how will I understand whether the DNA is inserted or not? I should have some proof, right? I just can't add the DNA to the complete structure and imagine that this has got mixed and then I insert it in the host cell. In order to avoid this and in order to avoid any kind of confusion or any kind of doubt, we need to have a full proof that yes, DNA is inserted in the particular vector. For that, we need the concept of selectable marker. Yes, that means if suppose this is my plasmid which is acting as vector. For example, let's take PBr 322. Yes, the most ideal one. Now, here it will have the ori region, fine, correct. Selectable markers means what? On this side, I should have some segments of DNA which act as markers. What kind of markers do you think they will be? Basically, for example, let's think it's here. Yes, there will be, since this is a plasmid in E. coli, there will be certain regions of genes which give unique property to the bacterium which is not exhibited by other bacterium. For example, in E. coli, you have the two drugs ampicillin and tetracycline resistance capacity. That means even if you grow E. coli in presence of ampicillin, that bacterial strain will grow in the presence of the drug. The drug will not be able to stop it because it is having genes for resistance against ampicillin. Yes, so ampicillin resistance gene. Similarly, somewhere other place in the complete genome, it will have tetracycline resistance gene. So, can I use these as my markers? Yes. That means what? That means if I insert my piece of DNA, yes, if I insert my desired piece of DNA in this portion, don't you think the complete gene for ampicillin resistance will get spoiled? Yes. It will get spoiled. That means what? Now this bacterium will not be able to grow in ampicillin medium. That means if I have a culture plate in which I have ampicillin and tetracycline added, those bacterial strains which will grow in tetracycline but not in ampicillin will have the PBr 322 which is transformed. That is how selectable markers are used. That is how the process is carried out in a foolproof manner. I should know selectable marker ampicillin had some kind of region where my DNA is added. That's the reason why it is growing on plates having tetracycline, because tetracycline gene is still there. It is still showing resistance, but ampicillin gene is disrupted. Correct? That goes with selectable markers. Whereas now these selectable markers should have proper recognition sites. Yes, they should have recognition sites. What kind of recognition sites? Imagine inserting the DNA in the host will require what? The first condition is nicking of the DNA. Yes, the host DNA or the vector DNA has to be cut to insert the transformed DNA. Correct, the DNA responsible for transformation. So now, in order to nick it, you will require what? Restriction endonuclease. In order for the restriction enzyme to work, the restriction endonuclease to work here, they require recognition sites. No, they are very specific. They will not go and cut the DNA duplex anywhere. They will cut it only at the specific site. So the selectable markers which you are selecting should be such that they have restriction enzyme sites also inside them. So basically your complete vector will have various types of genes. Yes, the complete blue circle that you see is actually having many genes. But what is of our interest is these two genes because they act as selectable markers. And these two selectable markers have several recognition sites for the restriction endonuclease to come, cut it in between and then the DNA to get inserted. This is how the complete cloning vector is formed. This is how you make it so that it is sure on your part, you are sure about it that the complete gene is inserted in the vector and now it will go and get transformed or it will transform the organism in which you are inserting it. Yes, so if you insert this in another E. coli, then you will have the E. coli making your product for sure because you know the gene is present in your E. coli. Now this was about the bacterium in general. What about plants and animals? In plants and animals, what kind of cells will make sure that you are getting the gene transformed? Basically, when you talk about plants, yes, we have the largest, the more number of experiments which are carried out in case of plants, the largest and the most widely used plasmid is your TI plasmid. Yes. TI stands for tumor inducing plasmid. This is basically from the tDNA, which is the DNA which is responsible for formation of tumor DNA that is present in the bacterium present in the soil called as Agrobacterium tumefaciens. Agrobacterium tumefaciens. This bacterium responsible for causing the crown gall disease in plants. Yes, sometimes we find tumorous growth on plants which you touch and then that forms powder. Yes, so that is caused by soil bacterium called Agrobacterium tumefaciens, which has this tDNA responsible for tumor formation. Now the TI plasmid which is taken and it is inserted in this bacterium such that this bacterium will instead of causing tumor in the plant, it will infect the plant but not cause tumor. It will transfer the genes which you have added. The TI plasmid will carry the genes that you desire. Yes, that will be present in Agrobacterium and now this Agrobacterium will infect the plant cells such that it will get the infection but not the tumor. It will get the genes that you have added. This is how it is carried out in case of plants. Similarly, in case of animals, you have certain viruses, certain diseases like your sarcoma virus and all which are transformed and then the animal cells are infected with it in vitro in the test tubes or in Petri plates. You infect them and you get the cells which are transformed. This is how the factors required for cloning vectors are studied. You need certain factors in the complete vector for it to act as a good vehicle to take your DNA inside. Yes, now let's meet up in the next lesson where we learn about the transformation of this DNA with certain techniques. Yes, suppose if I am not able to have a good vector, suppose if my genes are not fitting in this vector and I am not able to transfer the desired DNA into the genome of the host organism. So what next? I need to find out some strategies where vectors will not be used but you will still have transformation, may it be biological, may it be chemical, any kind of transformation of my DNA. Let's try talking about these kind of techniques in the upcoming lessons. Thank you so much.